Hello and welcome to chapter 16. This is our next to last chapter. We only have 16 and 17 left this semester. So you are almost to the home stretch. So in chapter 16 we are discussing the molecular basis of inheritance. So the molecular basis, the molecule we are particularly going to be studying, we now know is DNA. But we're going to start by going through how we figured out that DNA was that molecule that is inherited, and we'll get into a little bit about how it can be inherited. So in 1953, James Watson and Francis Crick introduced an elegant double helical model for the structure of DNA. So Watson and Crick are the two people who published this work. Most of their work was actually stolen from somebody else. They actually just walked into somebody else's lab and just swiped some notes off of another scientist, Rosalind Franklin's desk. So although she didn't get credit for this, because she was not a name that published it, she had died before they went on to get a lot of their awards that they won from it. Actually, Rosalind Franklin is a person who did the experiments to figure out that DNA is shaped as a double helix. A double helix means that, see, it's two strands together, so that's the double part, and helix, it's twisting. That twisting shape is what it means by helix. So here, see, this strand is twisting around, and this strand is twisting, so they're both twisty strands. So that's what it means, it's a double helix. That's just the shape it's in. All of our hereditary information is encoded in DNA. So because you are able to digest the things that you're able to digest, because you make freckles or don't make freckles, you can taste the things that you can taste, you can make the proteins that you make, everything you're able to do is because you have genes on your DNA. Everything you look like is because you have genes on your DNA, which can be affected by nature, so we won't get into that, but and then that DNA is what you pass on, what goes into your gametes and gets passed on to your children. Your DNA is what you inherited from your parents. DNA is reproduced in all cells of the body. Every single cell in your body has all the DNA of your body. So your eyeball cells have all the DNA. Your foot cells have all the DNA. Your stomach and heart cells have all the DNA. So your heart cells have the DNA to make tears, but it's turned off, right? Your eye cells are where that part of DNA turns on. And your foot cells have all the DNA needed to make heart muscle, but that is turned off in your feet, right? So you have all the DNA in all of the cells of your body, but in each cell you only turn on those DNA, those genes that you need for that part of your body. The DNA kind of leads this program to develop biochemically, anatomically, physiologically, and even to some extent behaviorally. So everything that your body is capable of doing can be traced back to genes on your chromosomes. DNA is copied during DNA replication. Cells can also repair their DNA, which is kind of nice because you don't want to have something damage your DNA and forever be stuck with broken DNA. Early in the 20th century, the identification of molecules of inheritance loomed as a major challenge to biologists. So that means that in the early 20th century, we didn't actually know what was the molecule of inheritance. There was a lot of beliefs that it probably was proteins, because proteins were a lot more complicated, and there were more amino acids. There were a lot more proteins in your cells compared to DNA that's just made of four nucleotides, and so it wasn't really seen as being that important. But Thomas Morgan, led a lab, and his group showed that the genes were located on chromosomes, and the two components of chromosomes are made of DNA and protein, became candidates for the genetic material. So once we figured out that chromosomes were being passed down, then what part of the chromosome was it? Was it the DNA, or was it the protein that the DNA was wound around? So first we did these experiments with Frederick Griffith. Frederick Griffith did these experiments, and he studied the role of DNA in heredity by studying bacteria and viruses that infect them. So one thing he did, he studied these two different strains of bacteria, and one of them was pathogenic. Pathogenic means one of them could cause disease, and one of them was harmless. It didn't cause disease. And so this was in 1928, so just about 100 years ago. So most of the people involved with this are not alive, but still this, on an evolutionary timeline, this is not that long ago. So here's these two strains of bacteria he was using. So he had this bacteria, this streptococcal bacteria, and it had this nice cell wall around it. Well, nice for the bacteria, because that cell wall made that bacteria, it kind of protected it from the immune system. So if that bacteria was injected into a mouse, or even if a human had that bacteria would be able to escape your immune system and cause pneumonia, and the mouse would die. It also was very dangerous to people. So those are living cells with the cell wall. However, if you took the cell wall off, the strains of this bacteria that didn't have, I'm saying this wrong, the capsule, they had a capsule, a sugary capsule around their cell wall. These R cells are the non-pathogenic. They are not going to cause disease. So you can see they're still living cells, but they don't have that capsule around it. That capsule is a sugar coat. So without that sugar coat around it, kind of like the cover of an M&M, you just take that cover off of it and you're just left with the chocolate inside. So without that sugar coat around it, these bacteria can't escape your immune system. So your immune system is going to find it, kill it, and you'll be okay. So you inject that into a mouse, its immune system kills the bacteria, and they're fine. Okay, these with the capsule, the bacteria kill the mouse because they escape the immune system. If you take these dangerous ones, though, and kill them, even though they still have the sugar coat on them, their capsule, the mouse is healthy. So it's not the capsule by itself, it's the capsule around a living cell, right? Because the capsule has to be protecting something. So that capsule has to be protecting living cells. Without a capsule protecting living cells, you just have the capsule by itself, the mouse is perfectly healthy. But here's where it gets interesting. If you take these dead cells that have a capsule, perfectly safe, living cells that don't have a capsule, perfectly safe, you put these two together, the mouse dies. So this doesn't kill it by itself, this doesn't kill it by itself, but put together they do. What is going on here? Why would those two together kill a mouse? And what they found when they took and autopsied this mouse is they found living S cells. So these two mixed together ended up with cells that were living and making a capsule. So somehow these non-capsulated cells were able to take the DNA or to take some sort of genetic material from these cells to make a capsule. They take that ability to make a capsule into themselves, and you end up with living cells again. That's pretty exciting. Like, I love this experiment because I think it's just so beautiful, how we're figuring out how, not only like how bacteria work and how things cause disease, but also figuring out like how do cells gain the ability to do things? How did these cells get the ability, these non-encapsulated cells gain the ability to make a capsule? Because they took that information from these dead cells. Somehow they took something. Did they take a protein? Did they take a DNA? What did they take? Today we know they took DNA from these cells. So whatever DNA these cells had, these living cells were able to just pull it into themselves, so they were able to start making those capsules. Okay, so this process is called transformation. A transformation is a change in genotype. So that change in genotype was they were physically able to do something new. They physically were able to make a capsule. It's a change in genotype because they took in genes, and a change in phenotype. They're physically able to do something new. And that change in phenotype and genotype was due to assimilating foreign DNA. They took in DNA that wasn't their own. They took in DNA from those other cells. Then work by Avery, McCarty, and McLeod identified that the transforming substance was DNA. And Avery, McCarty, and McLeod had some beautiful experiments. I hope we're able to talk about that in this chapter. But even after they did their work, a lot of biologists stayed skeptical about it because we really didn't know a lot about DNA before the 1950s. We really just hadn't studied DNA a lot. And then more evidence for DNA as a genetic material came from studies of viruses that infect bacteria. So these viruses are called bacteriophages. Sometimes they're just called phages. And these viruses do not infect people. They're very specific for what cells they can attach to. They only attach to bacteria. They only infect bacteria. And these viruses are used a lot in molecular genetics research and studying how genes travel. So to have a virus is not a living cell. It's just a piece of DNA, or sometimes instead of DNA, it'll have RNA, and then around it has a protective coat, just a protein coat. So here, this is DNA, and this is the protein coat around it. Bacteriophages have a little bit more complicated structure, that they have this, tail sheath and tail fibers. This greenish blue teal colored virus is attaching to this brownish yellow bacterial cell. And so it's attaching with these tail fibers, and it kind of acts just like a hypodermic needle. It's just going to squirt its DNA into the bacterial cell. So here's its DNA coming into the bacterial cell. And when it does that, that DNA is going to take over the bacteria cell and use the structures within the bacteria to replicate more viruses and make more viruses and spread around until it's infected other bacteria around it. So that's kind of how viruses work. Even viruses that infect people are still made by DNA or RNA with a protein coat around it. They'll inject into your body, into your cells, copy themselves, and then bust out and spread to other cells. So let's look at this phage. Again, what is a phage? This is a bacteria that can infect viruses. And we're going to look at how they reproduce themselves. Let me turn this up a lot so that you can hear it. And let's play that.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T2 phage, which consists of DNA inside a protein coat. The reproductive cycle of the T2 phage begins when the tail fibers of the phage stick to the receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes are used to manufacture phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall, and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the reproductive cycle of bacteriophage T2.
Okay, so by the end of that, it has killed a cell. So that's why virus infections, if you get an infection with the virus, you can end up feeling sick because your cells are actually being damaged. You're not going to be infected with a phage again, but the viruses that infect people will also kill your cells. You'll end up with an immune response to clear your body of that virus, but until you are cleared of that virus, you're going to feel sick because your cells are being harmed. Okay, so let's go back to what any of this has to do with anything. Okay, so in 1952, Alfred Hershey and Martha Chase, their story always makes me want to eat a candy bar because hearing that Hershey all the time. Okay, anyway, Hershey and Chase, they showed that DNA is the genetic material of this T2 phage. So they had this experiment, and in the experiment, only one of the two parts of the T2 would enter the bacteria. So either the DNA or the protein would enter the E. coli cell during the infection. And they concluded that the injected DNA provided the phage the genetic information. So in batch 1, they labeled the proteins with this radioactive sulfur. In this other batch, they labeled the DNA with this radioactive phosphorus. So they labeled the phages. These phages infect the cells. And then they're going to shake off the extra parts. So whatever is outside of the cell gets shaken off. So you're only left with the cell, with the bacterial cell, and whatever has gotten into the cell. All these little pieces outside are going to come off. And then they're going to centrifuge the cells, form a pellet, and then they're going to measure radioactivity. So if the protein is what is getting into the cell, you'll see a lot of radioactivity inside the cell. Here, if the DNA is what's getting inside the cell, they're doing the same thing again. Label the DNA instead. The cells get infected by the phage. They shake off whatever's outside, and then whatever is inside is left. And sure enough, here in these cells, the DNA is in the pellet. The DNA is what is radioactive in the pellet. So it's radioactive DNA. The DNA was found in the pellet inside of the cells. Here the radioactivity was found in the liquid because it was outside of the cell. So this tells you that the protein was outside of the cell. It was not getting into the cell. Here the radioactivity, because it's in the pellet, was inside the cell. It was getting into the inside the cell. So that tells us that whatever these viruses are injecting into the cell is DNA. So this is big information that the viruses are able to reproduce themselves through DNA, and that the viral DNA can program cells. So that was strong basis that the genetic material of viruses was DNA. That didn't mean 100% the genetic material of people would be DNA, but it was a very good indication that that was probably the path that we should be going down, that genetic information in people was probably also on DNA. Here's Hershey and Chase and their lab. This is what they did.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
Okay. So, once the scientific community started believing that DNA was a genetic material, we're going to start talking about studying how we studied what was happening in DNA. So DNA is a polymer. That means it's made of repeating subunits. And each polymer has a nitrogenous base. So here, these are the nitrogenous bases, T, G, C, and A. Those are the four bases we see in DNA. They have a sugar. The sugar here is this 5-carbon sugar, 1, 2, 3, 4, 5. This 5-carbon sugar, so it's a pentose. And in this sugar, one of the oxygens has been taken off, so it's called a deoxyribose, and a phosphate group, that's this PO4. That's the same kind of phosphate like we see in ATP or ADP. So those 3 parts are the 3 parts of a nucleotide, and that repeat. So you can see these same 3 parts in this next, and this 3 parts, and these 3 parts. So those 3 parts just repeat to make the whole DNA molecule. Nitrogenous bases are A, T, C, and G. And in 1950, Erwin Chargaff reported that DNA composition varies from one species to the next. So you can stop and see what is the percentage of A, T, C, and G in this animal, which is going to be slightly different from A, T, C, and G percentages in the next animal. So each species kind of has its own relationship of how many of each different bases there are. So this ability to see that there's different compositions of DNA, different species can have different bases, also help to make DNA a credible candidate for being the genetic material in animals. So, you know, remember by this time we pretty much were believing that DNA was the genetic material in viruses and probably in bacteria, but for more, you know, multicellular organisms, things like animals and plants, this was starting to really show that it was probably the genetic material also in us and other animals. So from Chargaff's work, he came up with 2 rules. So the first rule, the base composition of DNA varies between species. So because you know what your percent of thymine and guanine or whatever is in one animal doesn't mean it's going to match in the next animal. So base composition can vary. And the second rule, which is kind of important to what we're going to talk about the rest of this semester, in any spaces, the number of A and T bases is equal, and the number of G and C bases is equal. So if you have a cell and you know that there's 15% of the bases are thymine, then 15% of the bases are going to be adenine. So that's 30% of the bases are going to be thymine and adenine. That means that the other 70% is going to be guanine and cytosine. So it's going to be 35% guanine and 35% cytosine. Okay, right? And then that adds up to 100%. This was just consistent every time he looked at any species. A and T were equal and G and C were equal. Okay, so what if you have a species that is 20% thymine? How much is the rest of it going to be? Pause and write down how much is going to be thymine, guanine, cytosine, and adenine. Okay, if it's 20% thymine, how much adenine? It's going to be 20% adenine. That's 40%, so that means you have how much left? 100 minus 40 is 60. So you have 60 to split between guanine and cytosine, so it's 30% guanine and 30% cytosine. Okay, so that is a question you need to be able to tell me, you know, thymine, guanine, cytosine, adenine. If I tell you how much percentage of one base there is, you can figure out all 4 bases. At that point, Chargaff didn't know why this rule was always found, but it always was consistent. Once we understood the double helix and the structure of DNA, it makes much more sense why that happens. You already know our DNA is base pairing because we've been talking about this from the first week. So thymine and adenine are always base pairing. So of course they're going to be equal because whenever there's one, there's another. Guanine and cytosine are always base pairing. So of course they're going to be equal. Whenever there's one, there's the other one on the other strand. Okay, we're going to watch this video and then we're going to pick up with the second part of this lecture. Oh.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these three bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, I will meet you back in Chapter 16, Part 2 lecture video. See you in a little bit.